Hello, my name is Ed Chapman, and this video cast is going to cover the cell cycle. To divide or not divide? To divide, that is the question. Cells divide for one of three reasons. They either divide to reproduce the organism, for example, to make more individuals so that descendants can travel through time and keep a species going. Cells reproduce to add cells to a multicellular body. We know this is growth. And also cells can divide to replace or repair other cells that have been damaged. Most of the cells in your body, for example, in a human body, are not dividing. They're just doing their jobs. The job might be moving stuff around, making chemicals, transportation, muscle cells contracting, like in your heart, on and on and on. They're just doing their job, but they're not actively dividing. They may or may not be replaced when they wear out or when they die. For example, the cells in your head, in your brain, are not well replaced. Some of them are replaceable, but not as well as tissues or cells you might have in your skin or in the lining of your mouth or in your blood, your bone marrow. Those cells are dividing all the time and constantly replacing cells that are wearing out and dying. Others, like your nervous system, aren't replaced nearly as well.
Ava Bergen, if you're still in the building,
sorry about the announcement.
Office. Ava Bergen, please call extension 2500. Ava Bergen.
All right, some of the cells in your body have very long lifespans. They live for decades. Others may only live for a few days. Okay, now cell division, the basic job of cell division is to divide up chromosomes correctly, which means that each of the two cells that you get after a cell goes through cell division has a complete set of all the chromosomes that it needs to be normal. And this complete set is called the genome. The genome can't have any missing chromosomes, it can't have any extra chromosomes, and it can't have any pieces of chromosomes. Those three things can make a genome abnormal and cause problems. For example, the human genome contains 46 chromosomes. You get 23 from your mother, coming into you by way of the egg cell that we all started with, and 23 coming in with the sperm cell. Of course, egg and sperm together during fertilization gives you a complete genome of 46 chromosomes. These two sets interact with each other according to genetics to build you and make you and regulate you in the way that you were genetically programmed. We'll talk more about that later. Chromosomes all across biology are made up of two chemicals, DNA and proteins. The DNA part is where the genetic code is stored, and the proteins organize, wind, and protect the DNA and regulate it. This DNA-protein combination or complex is called a chromatin. When cells are dividing, the chromatin contracts in the thick, visible chromosomes that we're all used to seeing, those wormy-looking things. When cells are not dividing, and when the DNA is actually being used to do stuff, the chromatin is spread out. It looks a lot like coffee grounds under the microscope. So here's a picture. If you look right here, this is a cell here's the cell membrane. Here's the nucleus here in the middle, slightly darker. It's been stained with a red stain. I think it's eosin it's called. And this cell is in interphase, which is a way of saying that it's not dividing, and you can see that there's no visible chromosomes in here. But if a cell is starting to divide, as you can see in this cell here and this cell here, the chromatin is condensed into something that's more easily visible, and you may even be able to count chromosomes when the cell is dividing, but not when it's not dividing. Okay, before a cell divides, obviously, the chromosomes have to be replicated or copied, because how can you divide a cell and have two identical cells produced by that if you only have one copy to begin with? So some duplication has to take place here. So after replication, each chromosome is actually made up of two copies held side by side, and they're held together in the middle by a region called the centromere, and each copy, like here's one here on this side and here's one over here, each copy is now called a chromatid. The whole thing is still a chromosome, but in this chromosome it's replicated, so you have two chromatids. This one chromosome over here, you only have one copy. When you have two copies, you use the word chromatid or sister chromatids to describe these two parts. Okay? Replication cannot occur when chromatin is condensed.
Please come to student activities. Nina Panoni, please come to student activities.
Sorry about the announcement. All right. Now, cell division basically has two parts, the mitosis part and the cytokinesis part. Cytokinesis is the process that divides the cytoplasm between two cells. Okay? Mitosis is a process that separates the sister chromatids into two piles that will become the daughter nuclei. Each pile contains one complete genome, and each pile is as far as possible from the other. So at the end of mitosis and cytokinesis, you have two new cells, and each cell has a complete nucleus in it containing a complete genome. Plants and animals differ a little bit in the way they do cytokinesis. We'll talk more about that later. Okay, mitosis plus cytokinesis together gives you two identical cells. They're also known as clones, also known as asexual reproduction, and each of those clones is a genetic copy of the original. Meiosis, on the other hand, it looks a lot like the word mitosis, so be careful when you spell this. Meiosis is a special kind of cell division. It has a different job. It does not produce clones. It produces a special kind of cell evolved for reproduction called a gamete. Gametes are also known as sex cells or egg cells and sperm cells. And each gamete contains only half the number of chromosomes of the original cell. We'll talk more about meiosis later. Okay, now the mitotic cell cycle basically alternates between interphase and mitosis. So if we have a cell that's going through a cell cycle, it's going to go through mitosis, cytokinesis, and at the end of cytokinesis you now have two identical cells. Those two can then go through interphase and mitosis again to produce growth. Alright, so one turn of the cell cycle produces two identical cells. And if you keep this going, you can quickly see how this is an example of exponential growth. Each time the cell cycle happens, you double the number of cells. So this is one of the ways that bacteria, for example, can go from one bacteria to millions in a matter of hours. It's a very, very quick growth curve. It's got a steep slope that's constantly increasing. All right, now the cell cycle basically has two parts. It has interphase, which is the part of the cell cycle where the cell is not dividing, and as you can see in this diagram, it makes up most of the life of a cell. And then there's the mitosis and cytokinesis part. Combining these two together, you get something called cell division. So mitosis plus cell division gives you the whole, run that back by again. Interphase plus cell division gives you the whole cell cycle. Now interphase is divided into three parts: G1, S, and G2. And we're going to talk more about what each of those parts is in just a second. All right. Now a cell can be signaled not to divide. And if that happens, we say that the cell is in something called the G sub O, or a resting state. It is not going to be dividing. And like I said earlier, this is where most of the cells in your body are right now. They're not going through cell division. They're frozen at the G sub 0 state. Now, during the G sub 1 part of the cell cycle, the cytoplasm and all the things in the cytoplasm are being duplicated and copied and grown. The cell's getting bigger. It's growing to its maximum size. During the S part of interphase, that's where the DNA or the chromatin material is being replicated. It's being copied, and that means each chromosome is converting from a single strand into two copies held side by side, those chromatids we talked about earlier. And during the G2 part of interphase, enzymes are going and checking and making sure each of those duplicated chromosomes is, quote, perfect, unquote, and that any errors that are found are repaired. In a perfect world, it happens this way, but of course, there's always exceptions. Sometimes mistakes get through, can cause terrible things like cancer, or mutations, which almost always are bad, but rarely are good, and that's what pushes evolution forward. Talk more about that later. All right, now let's look at mitosis in close-up. So before we can talk about mitosis, we got to go back to the late part of the G2 part of the cell cycle and look and see what's happening. All right. Now, during the late part of the G2 part of interphase, something called the centrosome, which contains the centrioles, is copying or duplicating itself to make two centrosomes. And if you look closely here, we've got the original one here, and then we've got a new one right over here. It's harder to see. This animal cell has been stained with some fluorescent dyes to make all these parts show up. The chromosomes are in purple. The spindle fibers and other fibers are in yellow and red. Okay, and if you look at this late G2 cell, the nuclear membrane is intact. It doesn't have any holes in it yet. Okay, and the chromatin, if you look in here, it's all spread out. You really can't see individual chromosomes yet. We're too early. Okay, now prophase is the first phase of mitosis. And there, as you can see here, there's some very different looking appearance in here. There's a very different looking situation here inside the nucleus. So inside the nucleus, we can see that the chromatin is starting to condense into visible chromosomes. Excuse me. And these visible chromosomes are becoming thicker as the chromatin winds itself around those proteins. Okay, now the nucleolus is also disappearing. Remember, the nucleolus is the location in the nucleus where ribosomes are being produced. That all gets taken apart. It dissolves. And now in the cytoplasm, the centrosomes are starting to move apart, move away from each other. So you can actually see two centrosome centers here and look like little yellow dots, and they're starting to move away from each other. And the mitotic spindle is starting to develop between the two centrosomes. Prometaphase is the next part of mitosis, and in prometaphase, the nuclear envelope has, by the time prometaphase starts, the nuclear membrane has completely disappeared. The nuclear envelope is gone. And the spindle fibers, which are these fibers growing between the centrosomes, are able now to penetrate and interact with the chromosomes. And we're going to learn about two types of spindle fibers, the kinetochore fibers and the non-kinetochore fibers. Now in metaphase, the chromosomes have been lined up at the equator of the cell. So these kinetochore and non-kinetochore microtubules have created a situation where the chromosomes are now lined up as far away from the two centrosomes as they possibly can get. They've been pulled there by the non-kinetochore microtubules. We'll look at them a little bit more closely later. Now the non-kinetochore microtubules are lengthening and pushing against the poles, making the cell elongate along this axis here. Now during anaphase, which is the shortest of all the phases of mitosis, the kinetochore microtubules are actually pulling the sister chromatids apart, and they do that by shortening. And as they shorten, they're transporting the chromatids towards the poles. Now once the chromatids are completely separated from each other, we now start calling them chromosomes again. Telophase is the last phase of mitosis, and this is when new nuclear membrane material is being formed around the two piles of chromosomes, which are moved to the opposite poles, and mitosis is now officially complete. A cleavage furrow may then start to divide the cytoplasm and all the goodies in the cytoplasm between the two new daughter cells. Okay, we're going to pause there and pick up with our next video cast.